Hello, my name is Graham Kingswood, and I'll be presenting with Genevieve Ravel on biofilms in the home. Thank you.
It's not just us stuck in our homes right now. Bacteria are too. So my name is Genevieve, and we hope you enjoy our presentation on biofilms.
To begin the presentation, we'll be going over the basics of what is a biofilm, how and why biofilms form, and the structure of biofilms. Then we'll move into specific biofilms that are found in the home, look at a case study where biofilms are in bath toys, and then where do these microbes come from that make these biofilms, the consequences of having biofilms in your home, and finally how to treat and prevent biofilms. So first we'll be looking at what is a biofilm. In the most basic terms, a biofilm is a collection or colony of living microorganisms which are attached to a surface and each other within a slimy extracellular matrix. Many different microorganisms can make up and form biofilms, including bacteria, fungi, and protists, though for this seminar we will be focusing specifically on bacteria. The environments in which biofilms can occur is widely varied, such as in your mouth with dental plaque that builds up on your teeth, or in ponds with pond algae which grows on rocks and causes them to be slippery. There is one requirement to the environment though, that is it must be wet or at the very least moist in order for biofilms to form. There are many different bacteria which are able to form biofilms with no set characteristic or shape between them. There are the gram-positive rod-shaped bacteria, which make up the genus Bacillus, the gram-positive coccus-shaped bacteria, which make up the genus Staphylococcus, the photosynthetic cyanobacteria, which can be seen on the image in the left, which make up blue-green algae, which is often seen in ponds. There are pathogenic bacteria which are able to form biofilms, such as gram-negative E. coli and Listeria monocytogenes, which causes diseases such as listeria. There are also helpful and useful bacteria which form biofilms, such as Lactobacillus plantarum, which is an important bacteria in silage, as it plays an important role in the Ebden-Meyerhof pathway, as well as in the fermentation of many vegetables in processes which create kimchi and sauerkraut. There's also ones which are used in the creation of cheese, such as Lactococcus lactis. With the formation of biofilms, there are 5 main stages which are in a constant cycle of repetition. The first stage is the attachment of a single cell to the surface upon which the biofilm will form. On the image on the right, these cells are shown as the brown ovals. As the cell continues to replicate or other cells join alongside it, it forms a singular layer of cells, which then begin to produce the extracellular polymeric substance, or the matrix, which actually makes up the biofilm. As more cells continue to join, or the cells continue to replicate, a multi-layered colony begins to form. At this point, cell-to-cell communication through quorum sensing, as well as the more symbiotic relationships which happen between the cells begin to occur. As the colony and the biofilm continues to grow, it takes on the more mushroom-y shape that it often appears and grows taller, though environmental factors such as the shear force of the water against the biofilm determines how large it gets and kind of the shape it forms. Once it reaches a certain size, the cells continually break off and detach from the biofilm and then kind of move down and can attach again to another surface and begin to build another biofilm. Biofilms are formed by bacteria for several simple reasons based on how they benefit the bacteria which create them. First, they protect from environmental factors. Stressors such as lack of water or high or low pH would normally cause the death of bacteria, but when the bacteria is within a biofilm, it's no longer as big of an issue. UV light would normally cause dehydration and death of the bacteria, but due to the extracellular polymeric substance, which creates the outermost layer of the biofilm, this UV light is not as big of an issue. This extracellular polymeric substance also helps create a microenvironment within the biofilm, where high or low pH in the outer environment does not impact the bacteria as much. Secondly, protection from harmful substances such as antibiotics or antimicrobials, which would be used to try to destroy the biofilm, are no longer as threatening to the bacteria because they cannot penetrate the extracellular polymeric substance, and certain bacteria may help protect other bacteria from these antibiotics. Finally, there's a symbiotic relationship between many of the different bacteria within the biofilms. An example of this is how autotrophic bacteria work together with heterotrophic bacteria so that they may cross-feed and each have the nutrients that they require. The structure of biofilms are composed so that a symbiotic relationship between the bacteria which create them can exist and flourish. The biofilm begins with a base of the bacteria which produces the EPS as well as the matrix, and as the biofilm continues to develop, different types of photosynthetic bacteria such as cyanobacteria, diatoms, and green algae will begin to appear, as well as other non-EPS and matrix forming bacteria. These organisms within the biofilm form symbiotic relationships between one another. This relationship revolves around the heterotrophic bacteria supplying carbon dioxide to the photosynthetic organisms through cellular respiration, and these photosynthetic organisms use this carbon dioxide to grow and produce oxygen from photosynthesis. Additionally, other carbohydrates, vitamins, and organic compounds are produced during these processes and are used as nutrients by both the autotrophs and heterotrophs. A mature biofilm reaches a steady state of equilibrium within these processes. The composition and structure of the biofilm is affected by the environmental conditions in which it occurs. When there's little light available, the biofilm is made up of much less photosynthetic bacteria, and depending on what nutrients are available from the water and moisture where the biofilm occurs, such as nitrogen or phosphorus, it determines what bacteria make up the biofilm. Also, shear force of the water also affects the biofilm, as it determines how large and developed it can become. Now we'll be moving on to biofilms specifically within homes. As stated previously, biofilms do their best in wet, stagnant areas. This is true within your home as well. In cases where pipes have water sitting in them due to a tap not being used or a clogged drain allows water to stagnate, biofilms are likely to form. They are more likely to occur due to untreated water or undertreated water, but can still occur in municipally treated water. It all has to do with how long they're allowed to stagnate and just the other circumstances, as the more microorganisms that are in the water, the more likely that biofilms are to occur. They are most dangerous when they are allowed to not just be superficial, but get deep inside a person. This occurs when they're inhaled as a mist and are allowed to get into the lungs, when they enter an open wound, when they are used with a contact lens, or someone washes their eyes and they have a contact lens in with water, or when they are sprayed directly up the nostrils. Once biofilms are able to get within someone's body through the routes mentioned in the previous slide, they can cause many different types of serious infections, such as lung, blood, skin, brain, or eye infections. Listed below are several of the most dangerous types of bacteria which create biofilms, which can be found within your home. The most famous is arguably Legionella pneumophila, which was the causative agent of a large breakout within a hotel in 1977. It was found to be breeding within the hotel's air conditioning system. Stagnating water allowed for the biofilm to form, and then it was spread throughout the building as a mist. In total, it led to the deaths of 29 people and the discovery of this specific bacteria and a look at how it was able to form and its biofilm.
Hello, this is Genevieve. We thought it would be super fun to show you some biofilms around our home, and I've volunteered my bathroom to see if we can try to find any. So we're going to start with the sink. I'd say the most obvious place to go would be the drain pipe. And yeah, you can definitely see some slime growing on the sides there. Even I think on the edge of the drain itself, there's some pink coloring. It might be. Move on to the tub. I've already done you guys the courtesy of checking the toilet. There was nothing there. So this is pretty wet. It looks like there might be some staining on the sides, but it also could be just dirt residue. And then here's the shower itself. It looks like there might be some staining on it. It could be little bits of film, but not too bad. I'd say the sink was the worst though. Definitely some biofilm in there.
The most surprising case of biofilm contamination around the home that I had found was a study by a group of scientists in Switzerland who were examining biofilms that were forming in bath toys. So, for a bit of background, in 1998 there was a multi-drug resistant outbreak of Pseudomonas aeruginosa, which is a pathogenic, or in this case, bathogenic microorganism that happened in a children's hospital in Melbourne, Australia, that caused a whole bunch of sicknesses. And this outbreak had been actually linked to the sharing of bath toys. So, the aims of this study specifically were to characterize the microbial growth on real bath toys and understand what factors drive their development. And this image on the right here shows two cute little rubber duckies, seemingly innocent, but as soon as it's opened up, it's filled with gross, sticky biofilm. And here is how they conducted this experiment. They had collected a whole bunch of bath toys from random households, cut them open, and described the biofilm appearances, their microbial abundance, and community compositions. Now, they had also wanted to look at the formation of these biofilms under controlled conditions, where they took new bath toys and used them in clean bath water versus taking another set of new bath toys and then use them in dirty bath water. And this image at the bottom is actually one of the toys that they had collected from one of these random households. And you can see all of the slime that had formed on the inner surface, which is kind of gross and kind of scary considering a kid was playing with it. Let's take a look at some of their results. All of the bath toys that they had collected, as well as their control samples, had dense, slimy biofilms on their inner surfaces. On this black and white image at the bottom, you can even see how thick these biofilms had grown on top of the material. And this top image on the left is a scanning electron image of biofilm that had formed on these toys, where you can actually see some of the bacteria, and it looks like there's some streptococcus chains in red, and then the green colored film surrounding them is the sticky polymeric substance that gets excreted. What they found in terms of microbial coverage was that the real toys had about an average of 9,500,000 cells per centimeter squared. And turning your attention to the bottom graph, the real toys being represented by the blue bars, there's still quite a bit of range within that average, depending on maybe how clean that household might have been or how often the toys got used. The clean water control toys averaged about 5,500,000 cells per centimeter squared, and the dirty water control averaged 73 million cells per centimeter squared. And looking at the graph again, you'll notice the clean water control has significantly less bacteria present, whereas the dirty water control a lot more. Ultimately, this study had concluded that there were 4 main factors that had impacted the biofilm growth and composition. The first one being the flexible plastic material. The bath toys were all made up of synthetic polymeric material, which is actually able to absorb a lot of organic nutrients floating around in the water, and they can pass them to the bacteria living on the inner surface and support their growth. The second factor was bathwater quality. Dirty bathwater contains a lot more stuff and the molecules that the microbes may absorb, which was why there was so much more biofilm present on the dirty water control toys. Different tap water treatments may have also contributed to the amount of biofilm present on a bath toy, and given that each household was random, the treatments and sources of the tap water might have been different. The third factor that impacted biofilm growth was the concentrations of dissolved carbon, nitrogen, and phosphorus after a bathing event, where varying concentrations have a lot to do with which bathing products are being used. For example, the type of shampoo, conditioner, and soaps. The fourth factor was contamination from the human microbiome and external environment. Because when you're bathing, microorganisms sitting on the skin get washed into the water, including urine residues and sweat that especially contribute a lot of ammonia to any microorganisms. Okay, so we figured out where bacteria come from looking at the bath toys from the case study, but what about the other areas in your home? How are microorganisms getting into your sinks and other places? Well, there's three main ways humans can contribute to the microorganisms growing in your home, and that is through your skin, respiratory traces, and feces. Again, dead skin cells fall off of us every day, and microbes come with it. We also release them into the air by talking, sneezing, and coughing. And lastly, they're sent down our pipe systems through feces. Through all of these ways, a microbe can easily attach somewhere, and when exposed to flowing water, they can thrive. But unfortunately, it's impossible for us to avoid spreading them. Let's look at some of the consequences of leaving a biofilm to grow. Worst case scenario is that some biofilms may contain pathogens and can cause serious illness if they're accidentally ingested, just like we saw with the Pseudomonas outbreak in the Children's Hospital. Biofilms can also easily spread and become very hard to kill if left untreated, especially if they develop antibiotic resistance. Often they can also build up and clog drains and may also stain and pit household surfaces, which becomes quite costly to repair or replace. And the image on the right shows the spread of Serratia marcescens, which is a common biofilm that grows in the form of a pink slime in between bathroom tiles. But it can be easily removed with baking soda and a lot of scrubbing. This leads me to discuss the treatment and prevention of biofilms in general. To reiterate, we can't prevent microbes from falling off our skin or sending them into the air by talking, but we can prevent their growth by regular household upkeep. And this is the most important thing. Bacteria will get removed by cleaning, and therefore they cannot spread and cause problems. Hand washing frequently also protects you from microbes that could accidentally enter your body, just in case they're sitting on any surfaces. In a situation where you do come across any biofilm growth, they can be removed by scrubbing the surface with a metal brush, soap, water, and 10% bleach. There are also products that are commercially available to treat biofilm accumulation, and one example is the ASUM product, which helps get rid of biofilm that is accumulated in a hot tub. In summary, we started by explaining that a biofilm was a collection of microorganisms attached to a surface that were held together by an extracellular slime. It was beneficial that they formed this community for protection and to help each other grow by sharing nutrients. Next, we went through the 5 stages of biofilm formation where the critical requirement was a wet surface. Wet surfaces are especially evident in pipe systems, which is where most of the biofilm in your home is growing. But they can also be found in places you might never have thought of, like the rubber bath toys found in the case study. If biofilms are left to grow, they can be hard to remove and ruin household surfaces, but regular household upkeep is sufficient enough to keep them at bay. Again, my name is Genevieve, and thank you for listening to Graham and I's presentation of the biofilms found in your home. Have a great day.